Okay, so then in a later mission, the 2008 Phoenix lander mission went to the northern polar plains. And one of the first things you notice here when you look out of this image of the northern polar plains is you see these polygons. And these polygons are due to ice wedge polygons that are in the subsurface. And so one of the really great things about this mission was it had an instrument called the Wet Chemistry Laboratory, and it was a solution chemistry. It was a solution chemistry instrument that had ion selective electrodes that went after calcium, magnesium, and other and anions like chloride and nitrates. It also had a pH electrode and electrical conductivity meter. And so at this landing site, we had a loose unconsolidated surface soil, ice-free surface soil, and in the subsurface, we had the icy soil. And when we did the analysis with the Wet Chemistry Laboratory, the analysis was basically similar between both samples. And the way the analysis was conducted was 1 gram of soil was put in 25 milliliters of solution, and then was analyzed with the ion selective electrodes and the pH meter and the EC meter. And the pHs were on the order of about 7.7 for all the soils that they analyzed. They analyzed 3 soils. And one of the surprising detections was the perchlorate detection, in the amount of about 0.6 weight percent in the soil. The perchlorate was detected actually with the nitrate ion selective electrode, but the signal was so intense that if it was nitrate, it would have weighed more than the soil. So they knew it had to be something else. And so they had said, well, then it has to be perchlorate. And in this soil, actually the perchlorate concentration was greater than the chloride concentration. And the counter or the opposite is true in the Gale Crater soils, where the chloride concentrations is greater than the perchlorate concentrations. They determined that based on the chemistry that you see over here, the sulfate was controlled by magnesium and calcium sulfates, and that if we had evaporated these solutions, then these solutions would have resulted in the formation of a potassium, sodium, or magnesium perchlorate, along with sodium chloride, anhydrite, and kieserite. One of the things I want to draw your attention to over here is the conductivity, which was on the order of about 1.4 to 1.9 decisiemens per meter. And this is in a very dilute solution, 1 gram and 25 milliliters. And so I'm going to get to this later, that you can imagine if you did more of a saturated pace analysis, that this would have gone up much higher and would have indicated that your soils are pretty saline. Okay. All right. Now for the next few slides, I'm going to talk about analyses. I'm going to circle back to Gale Crater here. And I'm going to talk about the X-ray diffraction analyses that were done on these soils, as well as the evolved gas analysis. And so we analyzed the rock bed form with X-ray diffraction, as well as a few aeolian dunes that you see over here. And so when we look at the X-ray diffraction data is dominated by an amorphous component that is dominated by silica, iron, and sulfur, and with some aluminum. The remaining mineralogy consists of plagioclase, feldspar, olivine, pyroxenes, along with magnetite, hematite, calcium sulfate, anhydrite, little bits of quartz, and some ilmenite. And so when you look at this mineralogy, you get a feeling that it's limited in alteration due by the presence of all these primary mineral phases. All right, now we're moving on to the evolved gas analysis that was conducted on these samples. And so the way evolved gas analysis works is soils are basically put in an oven, and this is the sample analysis at Mars instrument, which has a little oven, and sample is put in there, and then the samples are heated from ambient temperatures up to about 870 degrees centigrade. And as the sample is being heated, mineral phases or volatile bearing phases decompose at characteristic temperatures that are diagnostic of that phase. And so then we were able to detect these evolved gases with a mass spectrometer. And so some of the target gases we go after are SO2, O2, CO2, water, and things like that. And so in this case here, when we're looking at evolved SO2 releases, we see that the SO2 releases are occurring at higher temperatures. And the temperatures that are kind of in this realm here are more consistent with iron sulfates. And then these higher temperatures up here are more consistent with magnesium sulfates. So this is indicating that these samples here basically consisted of iron and magnesium sulfates. The levels of sulfates that we're detecting by this technique of iron and magnesium are far lower than the calcium sulfate that was detected by the Chemin instrument that I just talked about earlier. So these samples are dominated by calcium sulfate, but they do have contributions from iron and magnesium sulfate. When we look at evolved O2 releases, these evolved O2 releases are consistent with perchlorates on the level of about 0.2 to 0.4 weight%, which is a little bit less than what we detected in the Phoenix landing site soils there in the Northern Plains, which was on the order of about 0.6 weight% perchlorate. Moving on to CO2, these evolved CO2 releases that occur at these lower temperatures are consistent with organic phases, and then actually all the entire temperature range is consistent with organic oxidized organic carbon phases. However, these higher temperature peaks that we see here could have contributions from carbonates like siderite. So iron-bearing carbonates can give off evolved CO2 releases here. The concentrations of carbon that we detected in these soils are on the order of about 1,000 to 2,000 parts per million carbon. And so any organic carbon phases that are in these materials are likely derived from meteoritic or magmatic processes, though we can't, unequivocally exclude microbiological contributions. Evolved NO releases were also detected, which is mass 30. And these are consistent here with nitrates that are probably sodium nitrates, possibly. There may be potassium nitrates, but very small amounts on the order of 0.01 to about 0.03 weight% nitrates in these soils. Okay, so now I'm going to change gears here a little bit and talk about possible pedogenesis at a very, I guess, incipient level on the surface of Mars. There are at 2 locations on Mars, we've detected cemented indurated soils that are observed about 2,500 kilometers apart. And these both occur in filled in small impact craters in Gusev crater and in Elysium Planitia. And here are the filled in impact craters right here that you see here. And so you can see here, they're kind of places where a lot of dust and things can accumulate and don't really get blown out. It's basically a zone of accumulation, where the soils up here, you can imagine where things can get deflated away. So you can imagine these materials might here be a little more stable and allow things to develop over time, or allow soils to form over time. And one of the things they notice when they dig trenches into these soils, here's where a rover wheel actually dug a trench in Gusev crater, and here's actually where they had a penetrometer that penetrated the soil with a penetrometer. And the things they notice at both landing sites is we have these near vertical walls after excavation, and there are these overhanging soil beds and clods. And in this particular soil, we actually have chemistry of these soils here, and they do show increases in sulfur in these soils, higher sulfur contents that are associated with magnesium, so most possibly magnesium sulfates. And so we have these overhanging beds, these soil clods that are very stable, and it suggests that there is basically a dur crust that's forming in these surface materials here. And the thinking is that there's probably thin water films, possibly mobilized salts to form these salty crusts over geologic timescales. And so when we look at a sort of a horizon here of the Elysium Planitia landing site, impact crater here, we note that at the surface, we have dust, we have sand, but then this is just below the surface here, a centimeter below the surface is where this dur crust horizon occurs. That, is this a BZ horizon? Everything else are C horizons. And of course, CR horizons here at depth. But the possibility exists is that maybe there's some incipient soil formation that goes on, but it takes a very long time, obviously, for this to occur. And this is about the extent that you have possible soil formation, at least in the contemporary on the surface of Mars. Okay. All right. Now we're going to change gears again, and we're going to talk about plant essential nutrients. Since this is Texas A&M, we're into agriculture, we're into plant growth, things like that. And so one of the things about going to Mars is, and some of the things that like Doug Ming has worked on and Harrison's working on, and other people working on, is trying to understand, is Mars soil something viable as a plant growth medium for growing crops in enclosed life support systems on the surface of Mars for future exploration, human exploration of the surface of Mars. And so, yes, Mars soils do have plant essential nutrients. The thing is some cases it may not have enough. In some cases, it might have too much. In other cases, it may be just right. And so, as we talked about earlier, here are the nitrate concentrations that we detected in the Mars soils. This looks like a pretty low number to me. So probably have to augment your nitrogen to Martian soils in order to grow crops. Phosphorus. There is phosphorus in Martian soils, most likely in the form of apatites. The trick is it available? Well, there may be some challenges there too, because as we indicated earlier, there are nanophase iron oxides in these soils, and so they may, have issues or create issues in complexity and making some of that phosphorus unavailable potentially. Total, as far as potassium is concerned, there's quite a bit of potassium, but again, is it available? Here's what the solution concentration, the water solution concentration that was extracted in that Wickel experiment that we talked about earlier. And here's the concentrations in solution here. I'll let the soil fertility experts tell me if this is something that would be sustainable for crops. How long would that last? It's hard to say. Here are the calcium concentrations and the totals, and then the concentrations that were determined from that Wickel experiment at that one soil that was analyzed in the northern polar plains. And then here's your sulfur concentrations. So these values might be a little on the higher side of things than what's needed. Looking at the micronutrients down here in green, this is what has been detected in the Martian soils. Mars, being basaltic, there's probably molybdenum, there's probably boron. Not sure if there's sufficient amounts, but it looks like, everything is there, but, just in proportions that may not be as desired. And so this kind of gets us back to what I was talking about earlier, the electrical connectivity. I think Mars soils, based on that analysis, could have a salinity problem. Here's this very dilute concentration here. Dilute analysis gives you this value. You imagine if you concentrate that, you might have something on the order of like this in a saturated phase possibly. So that's a pretty high, that's excessively high from a salinity standpoint. I did a quick calculation using those water extractable cation values and got a sodium absorption ratio of about 1 to 2. So I don't think there's going to be a sodic problem. So it's probably mostly just going to be a salinity problem for Mars soils that will have to be dealt with. Okay, so you're a budding Mars soil scientist. You want to get the raw data to start understanding on, what are the Mars soils telling us, what they can be used for, what other things they can tell us about the ancient Mars climate. And so this is where you can go to get some of the raw data. And the Washington University in St. Louis manages a website called the PDS Geoscience Node. And when you go to this website, this is where all the Mars data is kept, from orbiter missions and lander missions. And you can get your hands on the raw data. And from the InSight landing mission, NSL, Mars 2020, all of them. This is where you can go for your one-stop shopping for soil data. Okay, so in summary, Mars soils are basaltic, and they basically have a broadly similar chemistry and mineralogy across all landing sites. They're driven by aeolian processes that redistribute and mix soils across Mars. These soils do have an alteration mineralogy that indicate aqueous alteration has had a role in soil formation, either at its source or possibly in place. There is possible evidence of early stage pedogenic processes in a few locations. Mars soils have potential as a plant growth medium. Most plant essential nutrients are probably available, but there are challenges with salinity, low nitrogen, and possibly phosphorus availability. And Planetary Data Science Node Geoscience Node is where you go to get the data to start doing research on Mars. So that's the end of my talk.
Awesome. Thank you so much, Dr. Sutter, for presenting all of that. We are a little bit over, but I just wanted to give anyone a chance to ask questions since we got started late.
I will ask a question.
Sure.
So, in some of those photos, I saw maybe at the beginning of your presentation that it looked like there might be some aggregation. And what did you call that again? It started with
Um, I'm sorry. Did I stop sharing or?
I do not see your screen.
Right, right. I think I stopped sharing. Yeah. So let me get back to sharing again. Why? Well, okay. So I'm sorry. What was your question again?
It looked like there was some form of aggregation of some of the Martian regolith potentially, but I could not remember exactly what it was called.
I was basically calling them little clods, uh, small little peds, basically. So they're probably being held together by salts. If
Okay,
that's what you're asking, what's the
that
mechanism?
was my
That
question. Oh,
there's that duracrust at the near surface that we think is sort of has concentrated salts in it. So kind of like you'd have a duracrust in a soil in a desert soil, for example. It's very thin, on the order of 5 to 10 centimeters in thickness. Um, but yeah. Does that help?
yes, that answers my question. Thank you so much.
Okay.
All right. Thank you so much for that awesome presentation, Dr. Sutter. Up next, we have Dr. Pandy. Dr. Pandy graduated from Texas A&M University in 2022 in the soil chemistry program under Dr. Schwab. She now works she works at Johnson Space Center and is a member of the Lunar Planetary Institute. Dr. Pandy, are you there?
Yeah, can you guys hear me?
Yep, we can hear you. I believe you have to share screen.
Yeah, let me share my screen. Do you see my screen? Do
Yeah,
you
looks
see?
awesome. Thank you so
Okay,
much.
perfect. Thank you. Thanks, Harrison, for the introduction. Hi, everyone. Thank you for the opportunity to let me present at this conference. I'm here to talk about clays and natural resources that we've identified on Mars over years of orbital as well as in situ analysis, and how we can take advantage of all the data that we've collected in order to inform any future human missions on Mars. So the future of human missions to Mars is going to be a international collaborative effort, and the initiatives to actually start this future mission has already begun. So the European Space Agency, Canadian Space Agency, Japanese, and NASA have been working together for missions that are as large as this scale. And the first most initiative towards sending humans to Mars definitely is the Mars Sample Return Campaign itself. And the Mars Sample Return Campaign basically involves bringing back these samples that have been collected by the Mars 2020 rover Perseverance as we speak. And so this initiative in itself is very important, which will definitely confirm or help us figure out exactly what the composition of some of the Jezero crater samples are, and it gives us a much more in-depth opportunity to look at some of these samples in addition to what we have done so far with in situ rover analysis. But to think about sending humans to Mars would mean to understand all the necessities that involve keeping astronauts or expeditions safe. And so, a really general place to start is by understanding what are these life sustaining essentials. And the first thing you would think we would need is the deep space habitation itself. We will have potentially a module that can be shipped along with the load, the initial load, but over time, we would expect that we will need further building of habitations. But in addition to having a shelter, we would also require basic needs. So a interesting study done by Versus in 2016 showed that a single person can consume up to 10 pounds of water, 5.5 pounds of food, and about 2 pounds of air per person per day. So that would add up really, really quickly, just given the nature of the mission, the fact that it takes us a really long time to get to Mars, and for us to be able to do any significant scientific objectives, to meet some of our science objectives when we do send humans to Mars, you'd think that we have to sustain this mission for a really long time. And so it comes to the idea that we need to figure out exactly how much of each of these resources do we need. So the key issues with this requirement is that we have limited cost. There definitely limited time, because sending resources takes a long time. It takes about 6 months for us to even get from here to Mars. And also, we definitely have a limited carrying capacity. So the load that's involved in each mission is highly limited. And so we have to be very careful about deciding what exactly is going to be taken with us at the very beginning, and how we can maybe source some of the other required resources over time on Mars itself. So what I'd like to focus on today is the actual key role that some minerals that are present on Mars, that we've identified on Mars, might have in obtaining some of these resources that we think are key components for human missions to Mars. So we'll start with the propellant and water. The Sabatier electrolysis is what has been proposed as the main source of getting both propellant as well as water in space. This has been demonstrated on the ISS for water, for getting water, where they use exhaled CO2 from the astronauts themselves, and they use the electrolysis to convert CO2 with the presence of some minimum amounts of imported H2 to generate water. Now, on Mars, we would use a similar electrolysis process of water to generate hydrogen as well as methane. So this could be a very self-sustaining source of water as well as fuel, because fuel is important for us to be able to expand any of our expeditions. And so where clay minerals come into play with this type of application is that we've been doing a lot of nanoscale development or nanomaterial development using natural clay minerals. And so clay minerals can be or have been tested to be used as important components of these solid state photocatalysts that are extremely important in catalyzing this electrolysis process. Another thing that has been identified in the Mars design reference architecture, which basically looks at all the things that all the main resources that are important for a human mission to Mars, is surface power. And this document lists nuclear power plants and solar power plants as the main potential sources of power when we are on Mars or an extraterrestrial surface. And so even in this type of extended application, clay minerals can come in handy, because they will be key components when we're developing the required infrastructure for such a power plant. But additionally, we're also seeing some really advanced applications of nanoclays in solar cells itself. So having a means to apply these nanoscale clay minerals, which are quite prevalent on Mars, in solar cells, will also help us potentially maintain what we can travel with us. And another main thing is habitation, of course. There's been a lot of studies that have looked at the application of clay minerals in developing composites. Usually, so for Mars, we would expect it would be a monolithic habitat module. Potentially, we would convert one of the spacecrafts that we fly to Mars on as the starting habitat module, but then to expand for other space, we would require taking resources from Mars itself. So basically, we would be transitioning to ISRU, which is in situ resource utilization generated products. And clay minerals have been identified as a key component for cementitious application for soil stabilization, which is very useful for dust reduction as well as horizontal construction. So if you're making roadbeds or anything that would require stabilizing soils so that they would not move. And then we would also have the opportunity to take other type of clay minerals to cast porcelain and other specialized parts that would help in habitation development. Oxygen, as we all live and breathe, is definitely another extremely important component that we have to think about. And the MOXIE experiment, the Mars Oxygen ISRE experiment, is one of the first ever ISRE experiment that has been conducted on the surface of Mars itself using the Perseverance rover. This is also another electrolysis experiment that was done to produce oxygen using the Martian CO2, and it can be also done using H2O. So similar application as the propellant in water for clay minerals here, we could definitely be using them as a component of the solid state photocatalyst. And finally, for food generation, there's been plenty of research ideas looking into this where we could apply the regolith, but I want to say that just applying the regolith itself is not enough, especially when you look at the Martian regolith, which has a lot of toxins as well as is lacking in a lot of nutrients, as Brad just mentioned. It does need to be augmented. So having clay minerals as part of this growth medium is definitely going to help augment not just nutrient cycling, but it also improves structure and also hydrodynamics. Okay, so I wanted to give you guys an idea about what different types of clay minerals have been identified on Mars. These yellow dots in the graph on top shows you all the different locations that we have identified clay minerals through orbital analysis on Mars. And these clay mineral bearing units have been dated to range from 4.1 to 3.5 billion years old. You can see that they're usually aggregated in the southern area of the planet, and that has to do with the transition of, or that's what we hypothesize, is that there's a transition of Mars going from a more wet climate to a drier climate. So we see a lot of clay minerals in areas that have the older formations that were formed when Mars was more wet. And so in areas like right here in wait, can you see there we go. So right here where Jezero crater is, where the Mars 2020 rover Perseverance is, we see this massive cluster of clay minerals. This right here, Gale crater, is where the Curiosity rover is right now. Also shows a clay mineral dot right there. But there are other areas as well where we see clay minerals, such as Marte Vallis. In addition to these locations, clay minerals have also been identified in craters, usually crater walls, ejecta, and central peaks. And these type of clay minerals we suspect are possibly from diagenesis, but we definitely see clay minerals coming from sedimentary processes as well. So in terms of composition of different types of clays that have been identified on Mars so far, this is pretty diverse. We've identified iron, magnesium smectites, chlorites, kaolin groups, including kaolinite and halloysite, as well as aluminum smectites, serpentine, illite, and muscovite. This is a older, similar diagram, but it's a little older, but I like this. I like to show this diagram because it gives you an idea of occurrence. So the percentages here are not weight abundances, but more of how many times each of these minerals have been identified within this area. So you can see that even though we have seen aluminum-based phyllosilicates, the iron and magnesium phyllosilicates tend to dominate on the Martian surface. So most of the Martian clays we can expect are iron and magnesium based, which would mean that if it is a 2:1 clay mineral, we would have a trioctahedral clay instead of a dioctahedral clay. Now this here I drew myself. Thank you, Dr. Deng, for helping me get here. It basically shows you how dioctahedral and trioctahedral clays can differ. One main important thing that I want to point out here is in trioctahedral clays, a lot of the substitutions that cause a charge on the clay minerals occur in the tetrahedral layer, which causes this highly localized negative charge distribution instead of a substitution in the octahedral layer on the dioctahedral clay up here, which causes a more delocalized charge. The main thing that this causes in terms of what we can see is the shrink and swell potential of these clays. So monazite would swell a lot more because it has a delocalized negative charge, which allows it to incorporate a lot more water as well as different ions in between the interlayers. And this might not be true. So the idea here is to show you that because we're seeing differences in compositions in clay minerals between Earth and Mars, when we think about applications, any ISRU related applications, we have to consider what the compositional changes as well as structural changes would lead to in order to get better results. Okay, so how do we test our ISRU practices? We definitely cannot or have not received anything back from Mars yet. We do have meteorites, but they're really expensive and rare. So we move on to soil simulants, and this is something Brad touched on as well, and Harrison's working on as well. So basically, these are synthetic planetary minerals, materials with composition adjusted to best represent what we have observed on the surface. And earlier simulants were actually basically just crushed rocks, just crushed basalt. But now that we have better understanding of what the composition of Mars is like, we are making more effort and augmenting some of these commercially as well as, well, actually most of them are commercially available simulants to include components that we know for a fact exist on Mars and might be actually relevant to the ISRU applications that we are trying to test. What I'm trying to say with that is that it doesn't make sense for us to just try and grow plants on a crushed basalt, knowing that the plant has no chance of surviving given the changes in density as well as lack in nutrients. It would make more sense to augment the base material with something like clay minerals in addition to maybe other nutrients that we know for a fact that plants would require but are maybe indirectly available on Mars and can be sourced. So let's look at the application of clay minerals in human development on Mars based on agriculture. So as I mentioned earlier, we were talking about di versus trioctahedral clays. Another thing, in addition to the localized negative charge on these clay minerals, the other difference is that the trioctahedral layer has a hydroxyl group that is oriented perpendicular towards the interlayer. So its interaction with the cations in the inner layer is different than that of the dioctahedral smectite or clay minerals. So that's another thing that we need to consider when we talk about nutrient cycling in these soils that have been augmented with Mars local clays. So there are hypotheses that in early Mars, there were instances where nitrogen was fixed into the soil, most likely through physical processes like thunder, lightning. And so we suspect that if there was, knowing that there was aqueous movement, most of these nitrogen is more likely to stay within the interlayers of these clay minerals. So we need to identify where we could potentially find these sources of nitrogen. And when we do add potassium, given the very low content of potassium on Mars, we'll probably have to take potassium-based fertilizer ourselves, but we have to understand how these fertilizers will interact with the trioctahedral having the clays in our ISRU materials to test, we would not just like help us understand, but what the effects are of augmented materials in agriculture, but it also helps us understand how we can increase crop variety. Another thing that people are worried about when you send humans to space is this idea of a limited menu fatigue. So we have to make sure that the astronauts are not depressed by eating just the same food over and over again. Another thing that we need to understand is also the microorganism diversity, how we can not just sustain a diverse microorganism within the growth medium, but also keep it within a closed loop system so we don't have any cross-contamination. And basically, we have to apply all the advanced practices that we've already studied in terrestrial applications and adapt them for Mars based on the Martian growth medium. A more recent study by Eschler et al. Looked at the most latest available Mars simulant is the MGS-1 to grow some romaine lettuce, and they were still unable to grow. Even though they added a nutrient augmentation, they were still unable to grow any romaine lettuce in this material. First of all, they did not augment this material with clay minerals, but because MGS is much more representative, it does have perchlorides in it, and so they suspected that it's more likely a toxicity effect. So all of these things have to be taken in mind when we are developing ISRU-based studies. Infrastructure development is definitely another application for the Martian regolith. There's been multiple studies that have looked at additive manufacturing or 3D printing using regolith materials that are stabilized either through geopolymerization or you can even remove the chemical reaction and add heat or pressure to sinter and create structures. But the main thing that I want to convey here is that having clay minerals is a great source for silica as well as aluminum in these systems. So when you add any cementitious material that adjusts the pH above 10, you will see a massive influx of these silica and aluminum in the system, which creates this high strength amorphous gel, which is really important for getting mechanical properties that are required for infrastructure development. Ceramics is definitely something that's been used with clay minerals on Earth for a really long time and can be adapted with Mars as well. This is a really interesting study by Carl et al. That looked at like I said, the latest simulant MGS-1, but this in their case, they augmented it with clay content adjusted to 5%. And they were able to create these intricate shapes, not just through slip cast or potter's wheel, but they also tried to use extrusion-based 3D printing to get sintered or ceramics that were comparable to terracotta and porcelain. I'm running out of time, so I'm just going to go really quickly with this last couple of slides. Other applications also involve clay minerals. There have been quite a bit of advancement in the biological aspects. Apparently 3D printing or 3D bioprinting is the next thing to look forward to, where they incorporate even clay minerals, and the clay minerals have a tendency to improve the bioprinting capabilities, where they possibly will print skin and cartilage and other aspects that are required for having a sustained medical system in space. With that, I'll just go to my concluding slide. We have detected clays of a diverse composition all throughout Mars. And so this informs our future ISRU-based technologies. There's definitely a need for a single database that lists all the past efforts that have been already conducted for ISRU, because anyone who's doing ISRU based research, when you do literature review, you will clearly find out that there's been lots of pockets of research that have been done, but nothing that's sort of communicated between researchers. So if we are to send humans to Mars, I believe that we'll have to have a single database where all the ISRU technologies that have been developed should be accessible. We should be using machine learning technologies to advance our methods of developing relevant admixtures based on the Mars PDS database. And clearly, you can see that most of the simulants that were available in the past lacked clay minerals and certain other features that we think are more ISRU relevant. And so these either need to be redone or there's room for new research for ISRU-based studies in using more relevant and better simulants. Okay, and with that, please check out the clay chapter that I wrote on ISRU that this presentation is based off. It is due to publish, I think, end of the year. And thank you very much.
Awesome presentation, Aditi. Thank you so much. We're definitely going to be checking out your new chapter. Wanted to open the floor for any questions that the audience might have.
I have a question.
Hi, Julie.
Hi, I was just curious. So given that all what you talk about these, how accurate are some of these simulants? Because we're using some of these simulants in our research, and I'm always questioning how close are they to the real thing. So.
That's the thing. I feel like our concept of simulant has to evolve from what it is at the moment. You know, the whole concept of simulants began as creating a material that would be geologically relevant to what we've observed on Mars. I believe we need to go from that base idea to now, okay, if we are to actually use this for different applications, we will definitely change things. So we can't just use crushed basalt as it is. We don't do that on Earth. We don't just take soil as it is and apply for other things. We augment them. Even in agriculture, we add and we alter and we augment. So we need to have a similar approach, maybe more sustainable based selection for what you want to augment these materials with. And that would be based on what we've observed on Mars. So having clay minerals added to your simulant, I think, is a great idea. And not just any clay minerals. I would probably go with clay minerals that have been actually identified and are potentially easy to excavate on Mars. That would definitely give you an edge on any ISRU-based studies.
Awesome. Thank you so much. Any other questions?
Yes. I won. So at the end, you mentioned the possibility of using machine learning to come up with new mixtures, new uses for clays on Mars. How much machine learning and AI work has been done in this field already, if any?
None.
Okay.
There's room. There's definitely room for research. I believe right now there's a lot of interest within the geology side of things, where people are using machine learning to process a lot of orbital data, because orbital data can be quite large packages, where you want to deconvolute one particular aspect within the orbital database. Then yes, I have seen some research in conferences, presented in conferences, that use machine learning. Also, it's used for astrobiology and understanding like large scale results, something mostly related to mass spectral results. But no, not for ISRU, and I think it's definitely a room for someone to try it out.
Thank you.
All right. Thank you so much, Dr. Pandy. That was, again, an awesome talk. Up next, we have Professor Yujin Ding. He is our clay mineralogist here at Texas A&M University. He graduated with his PhD from A&M in 2001, and I think a lot of us have had clay mineralogy with Dr. Ding. So we invite you in your presentation.
Dr. Hall and also Harris, thank you very much for organizing this meeting. And also Aditi and Brad, it's very nice to hear your presentation. Actually, your presentation gives an excellent introduction about what I want to talk about today. And also very, very glad to hear Aditi talk about a lot of clays. As a clay scientist, I'm very delighted to look forward to your chapter and also to read what you have summarized on Martian soils now. So today, I would like to talk a little bit about my perspective from a soil mineralist point of view and clay mineralist point of view. Today, what I'm going to show some data collected by our students. So the two of them I wanted to emphasize a little bit here. And the first one is Jessie Atkin. She's a master's degree student. She's working on some lunar simulants. And Brooke is undergraduate student. She's working in my lab right now on some Martian simulants. Aditi and Brad has done excellent introduction about the need about in situ resource utilization, but I would also like to extend a little bit more for the applications for space agriculture. So here is one of the documents released last year from NASA. And what's great about this one, it give a very clear blueprint about how to sustain human presence and exploration through the solar system. They give us a great blueprint, but at the very end, I also want to say a few words about why, as a scientist in agriculture, what we should do, how do we get our ideas, inputs into this blueprint when they issue another new version there. So in this blueprint, they also mentioned several objectives. One of them stand out related to us is some of them will be related to human and biological physics and physical in this part. And another very important and directly related to us is applied science. So in this part, basically emphasize they need to characterize accessible lunar and Martian resources, get the scientific research data, and analyze potential reserves to satisfy science and technology objectives, and particularly mentioned in situ resource utilization. Not only USA, NASA, this government organization, but also internationally, Aditi and Brad have mentioned Europe, some other agencies, but not only in Europe, but also in Asia, in Japan, in UAE, we have a lot of efforts. They have great goals for space exploration and also for space agriculture and horticulture. Right, in UAE, which is one place we had the chance to visit in a desert environment, they was trying to establish a center for space agriculture. So when we talk about the space agriculture, and also actually we think about what NASA has done, you will realize actually they have done quite a bit. So at the International Space Center station, they have started to grow vegetables for the astronauts there. A couple of years ago, we had a presentation from Kennedy Center, NASA, and then in this presentation, she introduced about the veggie. So for as a soil scientist and clay mineralist, I'm particularly interested in what kind of media did they use to grow those lettuces here. Actually, you will find that they call it a plant pinot. The major material actually is calcite monazite. It's one of the calcite clay, just as Aditi mentioned now. So not only at the International Space Station, but also you have heard the news a few years ago, like China has tried to grow cotton, tomato, and also rapeseed and some other plants. They have introduced this system onto the moon, and also they were able to let the cotton seed sprout. So when I look in this picture here, I was thinking about, so in this tiny device here, in this micro ecosystem they used here, they put the food biocompound producers, but also consumers here too. In this tiny device, so where do they get oxygen? Where do they get the nutrient? Where do they get water? So all of those questions, of course, in this one, they can sound from the packaging materials on Earth and sent to the moon. But can we produce them in situ? Can we generate using the soil on the moon and also generate enough nutrients, enough water, enough air for night? So those some questions as research or for our students from this, some questions we can think about on the moon. Well, not only USA again, but also here and other agencies and not government, but university, we have started work on some simulants here. So for simulants, and Aditi has mentioned a few problems that Dr. Junior was asking that question. So I'm going to talk a little bit more today. And in this particular example, showed here in Netherlands, they just use Hawaii basalt. This one, they have claimed they can produce at least 4 types of crops for astronauts now. So for us, basically the question will be, is it possible to generate water, oxygen, both macronutrients and micronutrients with what we have on the moon or on the Mars? So that's the basic question probably everybody can think of here. If you go there, what are you going to do? The questions as I did mention, also Brad mentioned, we have a lot of rovers and orbiters on Mars, and the sample return will only happen to 2026. So even until 2026, we may not be able to get a lot of samples to really grow crops now. So let's come to the point why we need to use the simulants here. So here, as I did mention, there are a lot of studies or research have been done with Martian and also lunar simulants. So I will just want to highlight a couple of them. There's quite a new one just released this year, early this year. This group also used Mars Global Simulants MGS-1. They tried to grow sweet potatoes. But if you read the paper, you will find one thing particularly I highlighted here. If they put 75% of Martian global simulants here, and will significantly inhibit the sweet potato growth, there's some disadvantages here too. So this is one part of the more or less similar studies you will find out. There's an excellent review paper on those recent simulation studies with Martian simulants. If you are interested, I can share with you. And also here, basically, is Jessica's work. She finished recently on the guidance of Dr. Terry Ginchu and Dr. Pearson. So she tried to use lunar simulants to grow chickpea. So this is some of our artists' views about how the lunar base should look like. And basically, the idea is try to in situ resources to grow plants on the moon. So as I did mention, and also Brad suggests there are a lot of toxic compounds or also some nutrient deficiency. We cannot just use the lunar or Martian simulants directly to grow plants. So something she did basically use a vermicompost and also using the fungi FM, Abastula, mycorrhiza fungi, and also tried to grow a chickpea in her study. So she had 4 or 5 of different ratios of those composite with lunar simulants, and she used artificial light, basically used blue light and also the purple light at different time to grow them in a Mala land growth tent, and also he watered those daily. So I will just share some final results she gave to me. So all of the plants exposed to the regular simulants exhibit some signs of stress, including stunting and reduced leaf area and limited shoot branching. And he also found out in overflow treatments, chickpea can grow to mature and also demonstrate seed site revealing successful production in those lunar regular simulant concentrations. But she also mentioned the chickpea did not experience bolting in local even 100% of those simulants here. So she has some hypothesis, and if she's continue do some test to check out if her hypothesis is right or wrong, and you are welcome to join her if you are interested in lunar simulants. And also she did some aggregation test with the snake test. Basically, she found out with the inoculation is with AMF, and it can increase soil aggregation dramatically. So she had some other ideas, try to expand the simulants to include more, and also use microarrays, other stuff as amendment to enhance the performance of those in situ resources now. So she has a QR code, some of the sites for you if you're interested in, and I can share this with you to hook your guests up. But as soil scientists and criminologists, we also want to look a little bit deeper to those Martian and lunar simulants, particularly we're using agriculture as simulations. How do they perform? So we got a few simulants from the Exolis laboratory. I did mention the early generations for simulants basically crushed the basalt and that's it. But with the accumulation of new data on mineralogy, geochemistry, so another approach basically using the Martian global simulant approach, and they just mix different amount of minerals and try to reach the same chemical and mineralogy composition as the real ones found on Mars. So here we have 3 Martian simulants. One is sulfate rich, one is the clay rich, and one is just the global simulant, regular one. And also we have 5 lunar simulants. Here we have 2 lunar Mars simulant. One is the one, just a regular one, is the one is a crushing more, is a fine dust lunar Mars simulant. And here we have 3 other lunar simulants. One is the lunar highland simulant. Second one is the dust simulant. Again, the third one basically added 25% agglutinates into that simulant. So we have done some preliminary analysis to see what kind of mineralogy or chemistry, even how real they are, as Dr. Howe mentioned here. So there's some XRD pattern, and actually they did some excellent job when they have everything they claim to have. But when we checked on XRD and also found something they probably added into the simulants as accessory minerals such as talc, chloride. They are not reported in the data sheet. And here we have the sulfate simulants. In this particular case, I want to point out one is here, very strong pigment due to gypsum here, and there are some other pictures I will summarize in a slide later on. We also had a chance to check those samples on the SEM. This is one is a relatively lower magnification view. You can find all of those minerals they have identified in the data sheet, but also found something a little bit additional interesting. We have found some deltams in those simulants too. So if you find a deltam on Mars, that'll be a great news. But here there's a little bit of zoom in look. We have some iron oxide particles on this andesite. And here there's a little bit more close-up look about the deltams and also some iron oxides we have identified on the Martian global simulants, and that's one ice. Here is the clay-rich sample. It does have some 12 sodium, most likely sodium montrolite clay in this sample. And also here, but when we look at the aggregates in, probably also include some mica or illite in those samples as well. So from nutrient perspective, maybe excellent news for potassium source now. So if we haven't done quantification by ourselves yet, but if we just look at the mineralogy data and have put in a data sheet, we can summarize a few points here. Those minerals can be a water source if we cannot go to the pool to transport the water, or if we cannot mine water from the deep ground. There's some potential we can use clay minerals or use evaporize, basically those soluble minerals such as the magnesium sulfate, calcium sulfate. Particularly for this magnesium sulfate, it has 50% water in its crystal structure. And as clay scientists, you know, we have come up to 20% of water in there. But also in this case, we need to pay attention to, just as Brad mentioned, the salinity will be huge issues here. Gypsum may not be a big deal because gypsum is common mineral in the soils, but the magnesium sulfate is highly water soluble. So it can cause a high salinity issue. It's one of the major challenges we're going to have to face. Second, as Aditi and also Brad mentioned, the imbalance of different cations that not be favored for our plant to grow. So the next part about some nutrients or toxicity might be kind of issues. We think we have most of the micronutrients or macronutrients are present in those simulants. The question is, how do we make them accessible for plant to grow? And last question is, we have some heavy metals such as chromium, nickel, strontium. They are relatively high, and also have used the simple handheld XRF to verify they are high. So how do we try to remediate those toxicity? If can we do any treatment to that one or not? So there's some questions about in terms of lunar simulants. We also have analyzed both type of lunar simulants, basically on the dust and the coarse material, and have very almost identical chemical compositions. So we have found what everything they have claimed and they have, and also such as we have some of the ilmenite and andesite, the plagial class, and but in this case we may even have some talc in this example here, in addition to the pyroxenes they claimed. Right, so for those highland simulants, they're also very similar. But once I want to just show one of those aggregates they used here. So those basically this manganese steel have a calcium structure like they claim. So the interesting part about the minerals and those lunar simulants, they do not have clays in them. They do not have sulfate minerals in them. So there's from the mineralogy perspective, we will not be able to generate water from it. So what we're going to do if we're going to create the lunar basin there. So that's the part I would like to point out. In terms of nutrients, because they have very similar chemical composition as the basalt. So I think we have the same kind of nutrient toxicity issues as our Martian simulants. So we did a little bit more to monitor the pH of our simulants. I think for the lunar simulants, they're a little bit on the higher end and almost to 10. So this pH might be too high for our crop to grow. And also for the EC, rather much the salinity. So we check a little bit, the first one just 1 to 10 ratio. We found the Martian simulants, they have very high EC reading, much higher than the lunar simulants. So we did a bit more, just use different water solid ratios to measure the EC reading. So one we have 1-to-1 ratio, and this one is almost 10,000 microsiemens per centimeter, so extremely high. 1-to-5 is about 3,000 microsiemens. So if we compare the data with the salinity classification, we'll find out all of our Martian simulants belongs to either severely saline or extremely saline soils. So it does give us some kind of bit challenge. How do we deal with those high salinity issue? If we cannot bring water in to leach them out, how do we do that? Can we convert them to less soluble form, or even make them for some other uses? So last part, I would like to say that in this blueprint document, I think all of those objectives, ideas are given by engineers, by scientists, but probably didn't have much input from agriculture scientists here. So I would like to ask our students, our professors, our researchers at NASA, and also probably speak loud a little bit more loudly to the people surround you, and also make them aware. We have people here, we have technology here, we can do something to speed up our space agriculture in the next couple of years or decades now. So on the last slide, I would like to say, maybe not today, but 2050, if we're going to have this kind of workshop again, we might be able to host it on Mars. So welcome to Martian Garden. We have a self-sustained soil, plant, microbiota, human, animal ecosystem. Thank you.
Thank you so much, Dr. Dick. I hope that we have this on Mars in the future. That would be pretty awesome. Is there any questions for Dr. Ding?
I just have a, can you guys hear me? This is Brad.
Uh-huh.
Yeah, I just, quick comment. When I saw all the mineralogies of those simulants, it was hard to tell what the distributions of some of those, oddball minerals like talc and biotite and all that kind of thing.
Mhm.
And it kind of brings me to the question that Julie asked me in chat about, simulants and things. I almost feel like, I highly recommend people working with simulants, go take a hard look at the literature. And this kind of falls on with the DD saying, take a hard look at the Mars soil chemistry and soil mineralogy literature and get a baseline so you know what is on Mars. And then when you look at those simulants, or when you're looking to buy a simulant, you kind of see that, okay, they're lacking this or that. You kind of know what you're working with. And I almost feel like seeing all this, it's like, it almost seems like we kind of probably have to make our own simulants, to really get it right, in order to do what we're trying to accomplish. Because I feel like some of these simulants are really not really as accurate as maybe they're leading us to believe. So anyway, just a word of caution.
Yeah, that's a great.
Mhm.
Go ahead. Go ahead,
I
Eugene.
agree with you. I think simulants, unfortunately, we assume they're homogeneous. The major challenge for us, I think, when we do agriculture simulation, probably we need a large quantity. If you try to form some of simulants by yourself, which I do see some researchers in the synthesis their own simulants, will be very, very challenging. So
True. Good point.
The major sense is the quantity.
Yeah, that's a good point. I guess I'm coming from the perspective of like just soil chemistry or mineralogy experiments, but you're right. Yeah, large
Definitely.
scale. Yeah,
If
large scale.
if some of our students really want to do that, this can be a business for them to develop some real.
Yeah, yeah. You bring up a good point, Eugene.
And the other thing I was going to add to that is, as you grow plants in the thing, there is such a thing as weathering. We know that occurs. And so that's going to change the mineralogy also as you continue to use it. So it's not just a static question, it's a continuing question. But I think it's time we need to move on since Harrison's the moderator and he's now speaking. I thought I would introduce him. So Harrison Coker is our next speaker, talking about terraforming Martian soils for using aeroponic culture and composting to try to get those simulants a little bit more, the simulants a little bit more ready for plant production. So take it away, Harrison.
Thank you so much, Dr. Howe. Can everyone see my screen okay?
Yes.
Yes, we can see.
Awesome, thank you. Yeah, so super awesome discussion today. And I would like to present some of our more agricultural approaches to recycling organic nutrients and thinking about how, when we get to these extraplanetary bodies, how we might deal with some of the low fertility situations that we're coming into. So as we've been talking about today, in recent years, there's been some really amazing rover science that's been done. We've had the detection of simple organics and a more or less increasing amount of nitrate, which is definitely good signs of potential plant survivability in these environments. And we're also looking at different active surface hydrologies and an abundance of water that we might be able to utilize once we're getting there. So we're crossing our fingers and we're getting really excited about lots of positives that are approaching. But in general, it is important to remember that the Moon and Mars both have not had the biological secession and the decomposition of organic matter, nor the evolutionary trajectory that Earth has had, where plants have developed for millions of years. And so they are exceptionally low fertility, and we are not going to be able to just plant our plants into these minerals once we get there and expect them to survive. We're going to have to create some kind of modified shelters, because unfortunately we're not going to be able to just get there and throw some plants out and then start watering them. And on Mars, another one of the major concerns that we're going to face for biological life is the high abundance of perchlorate. Perchlorate is anywhere from 0.4 to 0.6 weight% of the Mars surface, and it's formed by chloride-bearing minerals that are it's most likely formed by chloride-bearing minerals interacting with UV radiation to essentially form these salts that are highly deliquescent, and they're probably grabbing a lot of water that is available and creating subsurface brines that are extremely toxic and are going to have to be remediated prior to us getting there. And so part of the goals of my research thus far have been to think about ways that we can recycle these organic nutrients from soilless systems that we're going to be using, like aeroponic systems, and then also thinking about how we might remediate some of this perchlorate toxicity and also get some positives from that, so that we can live in nice bubbles and other things once we get there that are going to be highly buffered in the atmosphere and whatnot. And so perchlorate is a very soluble salt. It is capable of being leached out of the different simulants. And so here I'm just looking at leaching cycles using clean water and perchlorate up to 2 weight% to sort of look at like a worst case scenario once we get there. And you can see that after 3 cycles of leaching, we're able to leach about close to 90% of the perchlorate from the soil. Unfortunately, this is very wasteful of water, and it actually doesn't solve the perchlorate issue. It's just removing it from the soil. And so this is not likely going to be an extremely great remediation strategy, but you can see that even with a simple washing, we're able to remove close to 50% of the perchlorate, which is more or less pretty positive. And so I've proposed the use of biological perchlorate reduction, which utilizes the perchlorate reductase, chloride dismutase, and a variation of those depending on the species of microbe, to actually utilize perchlorate as an electron acceptor and to break this apart to generate available oxygen, which is actually something that we're very interested in using both for human consumption and for fuels and other sources. What's interesting is that biological perchlorate reduction is ubiquitous across many microbes on Earth, and perchlorate is actually in very low abundance. And so we're finding that a lot of our denitrifying bacteria are also capable of this perchlorate degradation pathway. And so sort of just start trying to get my experiment together and look at how these incubations were going to work. I went out to grab some soils from the Blackwood Prairie in Central Texas, and again, prepared that 2 weight% perchlorate incubation for like a worst case scenario. And this should create a salt condition and concentration that is not really going to induce any microbial life. But what I found was that when I isolated some of the microbes from that Blackwood Prairie and adding them to these incubations, we actually got several that were doing and performing very well. So there were definitely microbes in there that were hardy enough to survive in this soil. And but for this perchlorate trial stuff, I was using JSC Mars 1 Martian simulant that was kindly given to us by John Gruner at JSC. But we're trying to do a little bit of follow-up on do some metagenomics on these microbes to see who they are, and potentially think about what the active genome in that is too, with some metatranscriptomics. But it's probably not very acceptable just to go out and find some random microbes. And so at 2 weight percent, I was not able to get any pure microbial colonies to reduce the perchlorate, but knocking it down to 1 weight percent, I actually was able to identify 4 different microbial species that I ordered from the USDA that are known perchlorate reducers that were capable of reducing perchlorate. And so our winner was the Pseudomonas chlorophorus, and it had the highest perchlorate degradation rate. Each of those rates are significant. And so we're selecting these to further look at how the rate of perchlorate degradation and how much oxygen is ultimately going to be produced by this interaction. I don't believe that perchlorate reduction is going to raise the EC of these soils. As we've noted today, they're very salty, because I believe that the chloride is going to form it's already a salt. I think it's going to form other salts. So it doesn't necessarily remediate the situation, but maybe some combination of this leaching that we're doing combined with this microbial perchlorate reduction can satisfy eliminating the toxicity of these Martian soils. And generating the oxygen is a major plus as well. So, and I forgot to mention, but these incubations were performed at a 30% gravimetric water content. So they were relatively wet, but not to the point where they were becoming anoxic. And the incubations were also performed in anaerobic chambers, where we supplied a pure CO2 at about 200 millibar. So the Martian atmosphere is almost purely CO2, and it is extremely trace. So we had to concentrate it to get these microbes to live. But even by just concentrating it to about a fifth of Earth's atmosphere, these microbes are still hardy enough to survive. So we think that these are probably pretty good candidates to think about further assessing. And so following up with that, the next stage of my research was to utilize some soilless systems, in this case aeroponics, to recycle some of the organic plant waste and the inorganic nutrients that are circulating in these systems, and fortify some Martian soil with that. So I was using MMS2. We've been discussing the veracity of simulants today, and maybe this wasn't the greatest choice, but back in the day, I was really excited. John Gruner did say it was okay, but you know, we could get better with some MGS1 or something. It's what we're moving to now. But basically, the idea was that we could grow our plants in aeroponics, which aeroponics are very important in low gravity situations. So on Mars, the gravity is about 3/8 of Earth. And of course, in space flight, there is microgravity. And so hypoxic conditions can actually occur around the root system from the low convective transfer of gases. And there's been numerous spaceflight studies and microgravity studies that have noted hypoxia of plants. And so the aeroponic system actually missed roots that are hanging in an environment, and then it recirculates that solution. And so kind of looking at our pictures here, we had a couple of different treatments where we grew some wheat, which is, you know, wheat feeds the world. It's been used on these simulants before. Might as well go ahead and try it out again for this study. We grew the wheat out to harvest, and when the aeroponic solution was sprayed to the plant roots in the bucket, it was actually drained through these filters that you can see down below. That were these PVC housings that contained either the Martian simulant or sand control. And then after it was done, we were able to harvest the plants and take the biomass that we weren't as interested in, and then actually compost that back into the Martian material. So the idea was, we already have to have these soilless systems, especially for space flight, especially if the soils don't work at all. We'll try to recycle everything that we have, because we're not just an infinite generator while we're there. Root systems don't play around. So this is my coworker's study. He's looking at root exudates of wheat. A very similar cultivar was used here. And you can see that they are constantly producing these organics that are modifying the soil environment at all times. And so we're really just trying to accelerate what is happening in the rhizosphere, or the area surrounding the root systems, by concentrating this across these Martian soils. And so you can see that came out a little bit blurry, but on the left in our aeroponic system, when we went to go harvest the plants, the control, which was non-circulating, so it was constantly resupplied by fresh solution, was slightly, did have more biomass than both our Martian and our sand treatments, but that makes sense because it was constantly being refreshed with fresh nutrient solution and none of that was recirculating. And you can see that the canopy height of the control soils was significantly larger by the end of the experiment when we harvested them, but that the Martian and the sand treatments were not significant from one another. So it doesn't look like putting the Mars material in the filter in the aeroponic system is going to impede the plant productivity, which is great. So then we went on to composting. So we basically took all that plant material, we sort of dried it down, ground it up, threw it in the MMS2, and we kept it watered so that there was some, funky composting and whatnot going on. So we can see that as the modification occurred, this was straight out of the aeroponic system, and then out of the aeroponic system, we went ahead and composted our plant material into it. We can see that there is some slight aggregation occurring in the modified material, but then when we go to compost it, we see all kinds of aggregates and other things forming. So as for the pH, it is extremely alkaline when it is unmodified. It's around a 10. After augmenting, it goes down, and then it comes slightly back up due to composting. The EC values, again, 2.8, that would be very saline. Most plants would have a difficult time surviving in that range. The aeroponic augmentation was able to leach a lot of those salts out, but when we went to compost them, ultimately it rose back up, but it's at more acceptable levels for plant growth. And then the cation exchange capacity was reduced slightly during the augmentation, but then it ultimately increased, not drastically, but some due to composting. But as you can see, the CEC of our Martian material is relatively decent. It does have the capacity to exchange nutrients and other things, especially compared to the sand, which is what we're, more of an inert control that we were comparing it to. And the sand relatively underwent the same sort of trends with pH and EC here, except the EC of the sand really drastically increased upon composting, which is interesting. And so kind of getting into some of the carbon and nitrogen data, I'm sorry to drop these tables on us here, but for total carbon of our Martian material, we slightly decreased on the aeroponic augmentation, which is interesting, meaning that we really did not catch any of our root exudates. They did not absorb to any of our minerals, but it wasn't significant. But it was massively increased when we went to go compost it, obviously. For inorganic carbon, we saw no differences from the aeroponic augmentation or the composting. But for permanganate oxidizable carbon, which is a more readily available source to microorganisms and whatnot, we do see a pretty massive increase when we go to compost. For nitrogen, again, we don't see major increases when we went to aeroponically modify, but the total nitrogen definitely increases when we go to compost. And I don't want to bore us with this big table, but the same trends are available with the sand. And so with the carbon and nitrogen data, there wasn't that much occurring with augmentation. So the total elements are determined by ICP. We digested these in aqua regia, which is a mixture of hydrochloric acid and nitric acid. And just very briefly, we see some in our Mars material decreases in boron, sulfur, and zinc, but we do see increases in copper, potassium, and phosphorus. And so these are good because phosphorus is majorly necessary. Potassium is a majorly necessary element. Boron going down is not necessarily the greatest because boron is pretty limited across most of the Martian soils from what we're seeing. But sulfur and zinc are in high enough abundance that it actually brought those down to better, more acceptable levels for plants. We did total extractable elements as well, but I didn't include that because there were too many tables. Oh, I'm sorry. I just interrupted it with the next RD. We'll get back to that in just a second. For extractable elements, we saw aluminum, potassium, and phosphorus increase. Again, great to see that potassium and phosphorus increase, but the boron was ultimately decreased. Again, we're looking at that boron situation, but it is a micronutrient. And so we did a little XRD, shout out to Chaiwei and Dr. Ding for helping us with this, but ultimately we saw the leaching of gypsum was the only major change. Gypsum is calcium sulfate. And so we saw that weather away, which is very interesting, and I have some kind of hypotheses on that. But we further were interested in that phosphorus absorption. And so we had a visiting scholar from Taiwan who was an expert in synchrotron. And so we did some Zanes in order to figure out how our phosphorus was speciated across these samples. So I was only able to look at our unmodified versus our composted here. And from that, I was able to take some of our ICP concentrations in both total phosphorus and extractable phosphorus and sort of just extrapolate these out to look at the different species of phosphorus that were available. So organic phosphorus was an extremely limited part of even the composted treatment, despite that it increased very significantly in both total and extractable phosphorus. So the phosphorus is really being bound up to these minerals. It is probably not plant available, and there's probably a massive saturation of phosphorus that's going to occur. So I think that it is extremely important to start projecting, especially as far as data science goes, just how long it or how rare phosphorus might become in this environment, and what we're going to have to do to fertilize these soils in order to be able to provide a good source for the plants. But ultimately, all of this is meaningless unless we take our soils out of the aeroponic system and we actually improve growth performance with them. So I am performing a growth trial now with these different media here. I've been having a lot of issues with like pot size and other things with the wheat, especially with the small amount that I generated from my sample. But we are repeating the growth cycle multiple times, and what we can see is that the untreated MMS2 plants are dying around 3 weeks, and they are not able to survive beyond that period. But our modified and our composted Martian soil is actually competing very close to our potting soil and better than our sand environments. And so you can kind of see some of that here. This is sort of in just like the first couple of weeks, but our unmodified Martian material is here. It doesn't grow. It pretty much just tends to die. And then our augmented Martian soil is here, where the plants are actually, pretty much thriving. And the sand, it's interesting because our composted sand is not really performing as well. And I think it's because it is so nutrient limited. And it's also very salty. And then the potting soil is, of course, our positive control that's doing really well. But in the course of these different growth trials and like having growth chamber experiments go wrong, we get to pull some of these plants up and just like look at the rhizosphere of these plants. So for minerals that have never really been acted upon by biology, this is amazing that plants are exuding these materials. They are attracting microorganisms, of course, because we're not really in sterile environments here on Earth. And it is really terraforming these soils. So we expect that, the pH is definitely going to decrease as it does with rhizosphere, and that that augmentation and other things that are occurring will be accelerated once we're able to get plant life into there. But for a conclusion, we were able to find some different microbes that were able to biologically remediate perchlorate, and that we're going to do some evolved gas analysis to determine if oxygen or ozone or other volatiles are being produced. With our aeroponic augmentation and composting, so the total recycling of a system that we're taking up, we were able to increase total and extractable phosphorus, potassium and copper, and carbon and nitrogen. And so we're going to, this technology allows us to reduce the need to import fertilizers, and but we're still going to need to acidify the soil more because they're alkaline. What we saw from the pH of these soils was that they actually increased upon composting after they had decreased due to the aeroponic weathering. And so what's interesting is that I think that there was enough exogenous calcium and magnesium available that when we incorporated that organic matter, its decomposition produced a lot of CO2 that might be forming carbonates to further buffer an alkaline pH of these soils, which might be something that we're interested in because most plants tend to perform better at slightly acid conditions. And ultimately, we're probably going to still need to import our micronutrients, but in very small quantities. And so sort of further looking into this research, we're very interested in knowing what are the different rates of organic materials that need to be amended into these soils to make them suitable. This is MGS1, so we're moving away from the MMS2. But knowing what and how much organic matter on a carbon basis so that we can sort of normalize and actually provide recommendations that are meaningful for fertilization is super important. many of the studies right now that are growing plants in these materials are mixing organic amendments at volume ratios, say 30% organic matter to 70% mineral. And that's not exactly a repeatable method considering that our organic matter sources can vary widely. But that's all I have. Thank you so much for listening. And if you have any questions, I'd be super happy to answer any.
Thank you, Harrison. Are there any quick questions? I know we're a little behind schedule. We started a little late for those of you who came later. Any quick questions?
Harrison, that's a lot of soil in the back of that truck. Is that the Blackland Prairie soil?
Yeah. No, that was actually just down the road on Agronomy Road. We had to get some sandy stuff for some microbiomes experiments we were doing.
I guess, okay. So regarding the Blackland Prairie soil, why did you choose that soil? Are there native perchlorates in those soils? Because I know out in West Texas, there's some areas where I think perchlorates, their concentration kind of are higher than other soils. Is that why you chose that, or?
Yeah, so it was really just to try all the different perchlorate quantification methods because it's been difficult for us to do that well. And so I was back home, that's where my parents live, and I just grabbed some soil and was like, there's a lot of microbes in here. Let's see if any of them can reduce this perchlorate. Because for some reason, perchlorate reducing bacteria are ubiquitous throughout Earth's soils. So I just decided that we could start there. But I think it's proof that microbial resources are widely distributed across agricultural soils, and that as we're thinking about heavy metal toxicity and other machinery that we want, that these microbes are really capable of doing that stuff.
Okay. All right. Thanks.
Hey,
Thank you for your
Harrison, I have a
question.
quick question, if there's time.
Of course.
Yes, first of all, great presentation. Very,
Thank
very good to see. How many of these microbes do you think you will be needed to remediate a certain amount of perchlorate per soil? Meaning that, I imagine might be a pretty high amount of those microorganisms.
Yeah. Thank you so much for that question. So that's definitely very important to know. When I received these samples from the USDA NRRL, they were basically just these dehydrated capsules that you can hydrate with water and then kind of spread them out with nutrient solution. So I'm not actually sure the amount of microbe that I added, but in these follow-up experiments, that's definitely super important to do some colony counting. Unfortunately, none of these are culturable, so I'm not exactly sure how we're going to know that, but probably figuring out the least, based off of a dilution or something, maybe the least amount of microbe to add is super important, I would think. But yeah, thank you for your question.
I'd hope they would multiply,
I would
at
hope,
site.
yeah, I would hope so. I don't know what it is, but what's interesting is that like the perchlorate degradation pathways are unknown. They are not super well studied because perchlorate isn't really a major problem on Earth, but hopefully we can sort of maybe get into these mechanisms and whatnot of what is doing this. So I don't know.
All right, Harrison, I think we need to move on. I'm going to switch it back to you as moderator, and we can continue.
All right, thank you so much. All right, so up next we have Dr. Borja Barbero from Dr. Shippen's lab in the Biochemistry and Biophysics department at Texas A&M. Dr. Borja is an expert in telomere biology, and he's a super cool guy we've been working with a lot. So really excited to see what he has to present today.
Yes, awesome. Thank you, Harrison, for that kind introduction. Can you guys see my screen?
Looks good, Borja.
Awesome. So as Harrison was saying, I'm Borja Barbero Barcenilla. I'm a postdoc in Dr. Dorothy Shippen lab here in Biochemistry and Biophysics department. And today I'm going to be talking about growing beyond Earth, telomere tails of Arabidopsis thaliana, a lunar regolith simulant, and on the International Space Station. So as Aditi, Professor Deng, and Harrison have said masterfully, within the next decades, we're going to be seeing the colonization of both Moon and Mars. And we know that plants are going to be a critical element for all of those space travelers. However, we do have a major problem. We actually really do not know if plants are going to be long-lasting in space. So there are some key indicators of organism survivability and prosperity, such as DNA damage markers, epigenetics, mitochondrial dysfunction, and telomere homeostasis. So today I will be talking about telomere tails of Arabidopsis thaliana on the International Space Station and as well on lunar regolith simulant. But first of all, I need to remind you, what are telomeres? So telomeres are DNA repeats that are at the end of the chromosomes that they do not code for protein, and their main function is the protection of the whole genetic material. So if you the best analogy to explain what telomeres are, telomeres resemble the very tip of a shoelace, which is called the aglet. That aglet is in charge of protecting the rest of the shoelace, very similar as the telomere is in charge of protecting the rest of the chromosome. So in a very special importance is an enzyme called telomerase, which is in charge of adding these telomere repeats at the end of the chromosomes. In addition to having a major role in chromosome M maintenance, telomerase is important and has been associated in response to oxidative stress, as well as mitochondrial stability. So telomerase has been found in mammals to have roles beyond chromosome ends. So every organism has a different telomere length set point, which maintains the rest of the genome protected. So in Arabidopsis, Columbia ecotype, which is our model organism, that ranges between 2 to 5 kb, in mice 20 to 100 kb, and in humans 5 to 15 kb. So mutations in either telomerase or telomere-associated components can lead to changes in telomere length. There can be either telomere shortening or telomere lengthening. So telomere shortening can lead to early senescence, while telomere lengthening is one of the hallmarks of cancer cells. So the selection hypothesis dictates that organisms with short telomeres have lower chances of species prosperity. So in addition to mutations in either telomerase or telomere-associated components, environmental and oxidative stress can have an impact on telomere length and telomerase activity. So telomere dynamics can be influenced by a very big range of physiological and environmental factors, such as infection, inflammation, diet, exercise, smoking, psychological stress, environmental exposures, and of course, spaceflight. So all of these factors can lead to an increase in ROS, and in the CPEN lab, we have further tested that telomeres accumulate 100 times more ROS than the rest of the genome. So telomeres, you can think of them as sponges of reactive oxygen species. So why is that? Why telomeres accumulate more ROS than the rest of the genome? Well, telomeric DNA has a high G and T content, which they are especially prone to oxidation. It's important as well to mention that every organism has a complex antioxidant machinery that can help reduce the levels of ROS. So to study the effect of spaceflight on telomeres, there has been 3 major key astronaut missions. Twin study experiment, the 6-month astronaut mission, and most recently Inspiration 4. So in these 3 experiments, it was observed that astronauts aboard the International Space Station, they suffer telomere lengthening when they were aboard the International Space Station. This telomere lengthening was actually independent of the time that they were in the International Space Station, as telomere lengthening has been observed after 3 hours of flight. So one of the major hypotheses of why telomere lengthening could be happening is to an increase in telomerase activity, since human telomere length is directly correlated with telomerase activity. However, there was no telomerase activity measurement for in-flight organisms, as the protein was lost in space. So in addition to these telomere length phenotypes, astronauts did experience an increase in ROS, as well as genome instability and mitochondrial instability. So to test the effect of spaceflight on telomere length and telomerase activity in Arabidopsis thaliana, we plated Arabidopsis thaliana Columbia seeds on MS media at Kennedy Space Center. They launched on SpaceX CRS-22 on June 3rd, 2021. Seedlings were grown on the Veggie hardware, as Professor Den was mentioning. Here you can see the Veggie hardware with astronaut Thomas Pesquet inserting the MS place into the hardware. The plants grew for 12 days aboard the International Space Station, after which astronaut Mark Van de Heydt collected the samples, and then Megan MacArthur froze the samples, and then they were sent back to Earth. So as you can see, looking at the pictures from the ground control versus spaceflight, the roots are all over the place in space from samples. This is because there's no microgravity. There's microgravity in the International Space Station, so the roots do not follow any gravity vector to growth. They just follow light, so the roots are all over the place. So our first question was, what was the telomere length for spaceflown Arabidopsis? So for that, we measured bulk telomere length in both shoots as well as in roots, since we know that both tissues have displayed differences in spaceflight. And as we observed, there was no change in telomere length in both shoots and roots, as the control samples had a telomere length range between 2 to 5 kb. Remember, this is the range of Arabidopsis thaliana columbia ecotype, and a very similar telomere length range in space-flown samples. We quantify this here, as well as in roots. So no change in telomere length in space-flown Arabidopsis thaliana compared to the ground controls. We further tested this by looking at the individual telomere length, and the take-home message as well was that we observed, again, no changes in both shoots and roots in telomere length in Arabidopsis thaliana samples that were flown to the International Space Station. So this might surprise some of you, especially after I just mentioned that telomere length in humans was increased when they were International Space Station. However, we previously have seen in the CPEN lab that telomere length was not changed when our plants have been treated with other stressors, such as constant light, DNA damaging agents, high temperature, flooding, drought, as well as now microgravity. So in contrast to human telomeres, plant telomeres are remarkably stable in the face of different environmental stressors. So in humans, as I just mentioned in the introduction, telomerase activity is directly correlated with telomere length. So we hypothesized that we will observe no change in telomerase activity either. And to our surprise, we observed that the space-flown samples had a higher increase in telomerase activity in shoots to around 4x compared with the ground control, and a remarkable 150-fold change in space-flown routes compared with the ground-controlled routes. So telomerase activity was greatly increased in shoots and even farther in routes. So we, as I mentioned in the introduction, telomerase has been shown to have roles beyond chromosome ends in other organisms, and it has functions under oxidative stress. So we hypothesized that there could be an increase in oxidative stress and genome oxidation in space-flown samples. And that's exactly what we observed. We observed that both shoots and roots space-flown samples displayed an increase in genome oxidation compared to the control sample. So oxidation of the plant genome was increased during spaceflight. So the next question that we have is this telomerase activity increase necessary to protect the plant from genome oxidation? So we raise artificially the levels of telomerase activity here in the CPEN lab by having different mutants that have increased telomerase activity. And what we observe it is that telomerase activity is inversely correlated with genome oxidation, meaning that the higher telomerase activity the plants possess, they display lower levels of genome oxidation. So the next question that we possess was, okay, there's many factors that happen in space. There is microgravity, there is radiation. So what is leading to these changes? So we observed that simulating microgravity did not lead to any dramatic changes in telomerase activity. And further beyond microgravity, we have seen that other Earth environmental stressors that lead to oxidative stress, lead to telomerase activity upregulation in plants without changes in telomere length. So now we're hypothesizing that the space radiation is leading to increased oxidative stress. Increased telomerase activity is necessary to deal with this increased oxidative stress. So future directions, and in less than a month, we're going to be testing the effect of space radiation by taking different Arabidopsis thaliana mutants to Brookhaven National Lab. And hopefully by the end of this year, we will fly back to the International Space Station, in which we will bring telomerase mutants up to the ISS, see if they are better adapted compared with Columbia plants. So I just mentioned to you that telomere length remained unchanged in low Earth orbit. There was a telomerase activity increase that we hypothesized might be due to ROS. So in 2022, Annalisa Paul showed the world that plants can actually grow on lunar regolith, on genuine lunar regolith. However, these plants did display increased ROS. So if we are to grow plants on the moon, we're going to need to make sure that plants can grow through different generations in the lunar surface and in the regolith. So to test if plants could actually grow for different generations, we tested germination of Arabidopsis thaliana by bioremediating and washing of the regolith with different solutions. This was work by my former undergrad and former undergrad of the CIPEL lab, Emily Hall. And we tested different solutions. We tested untreated LMS1 was not able to germinate, which led to why we need to treat our regolith simulant LMS1. We used an antioxidant cocktail that led to a much better improved germination. EDTA and chelation did not lead to improved germination, and neither did the waste solution. So the old technique of growing potatoes following the Martian recipe did not really work for us in the CIPEL lab. But we did find out that the antioxidant LMS1 could be a suitable medium to grow plants in the lunar regolith simulant. So very similarly to Annalisa Paul's data, untreated plants grown on LMS1, they were able to germinate. However, they displayed arrested development past 25 days, and very nicely, and to our surprise, treated plants with antioxidant cocktail, they were able to grow in LMS1 and they were able to flower, and the plants did display a much better total size, as it was evidenced here in rosette area, as well as biomass, compared with the untreated. However, these plants are still smaller compared with our Earth control. But if we're going back to the moon, we cannot really depend on plants shipments happening every so often. We're going to need to be able to grow our own plants on the lunar surface. So what is the phenotype of continuous propagation of these plants in lunar regolith simulants? So as you can see over here in this picture, plants, G2 lunar grown plants, they are smaller compared with G1, which in turn they are smaller compared with Earth. So we quantified all of these plant physiology traits over here, looking at the rosette area, looking as well as the biomass, as well as the plant height. So there is a worsening phenotype of continuous propagation of the G2 lunar plants, even with having an antioxidant cocktail added into the lunar regolith simulant LMS1. But the next question that we have is, why do we see this worsening of phenotypes? What is happening molecularly to these plants? So what we did observe is that these plants did observe increased genome oxidation. So G1 plants and G2 lunar simulant grown plants displayed an increased genome oxidation compared with the Earth control. So even with antioxidant cocktail added into it, genome oxidation is significantly elevated in lunar grown plants. So as I mentioned to you in the introduction, increased ROS can lead to changes in telomere length. And we actually, to our surprise, we observed that wild-type Arabidopsis plants grown on lunar regolith simulant displayed a decrease telomere shortening happening over different generations. So telomeres are shortened when plants are grown in LMS1 for multiple generations. So there is a 500 bases loss in every generation of lunar grown plants. So you might be wondering, okay, what about if this phenotype is caused to a decrease in telomerase activity? And indeed, we observed that there was a decrease in telomerase activity in lunar grown plants to around 50%. However, previous research from the CIPEL lab has shown that plants that are haploinsufficient, plants that possess 50% of telomerase activity, they are able to maintain the telomere length at a normal range. So even though telomerase activity is reduced, we do not believe that this telomerase activity is leading to this shortening. Instead, we hypothesize that the excess genome and telomere oxidation is leading to telomere erosion, and this is leading to decreased survivability. So the word of caution here is going to be that genome integrity is going to be compromised with continuous propagation in lunar regolith. Okay, Borja, so you have shown me that plants are having telomere shortening when they are grown in one regolith simulant, so we went ahead and tested the same phenotypes using different ecotypes as well as different regolith simulants. So we use HOB-10, which is Arabidopsis ecotype grown in Sweden. And using this ecotype and using different regolith simulants, we observe the same phenotype in terms of telomere length. There is a telomere length shortening that is happening in every single regolith simulant that we have tested in the CIPEL lab. As well, we use another Arabidopsis ecotype, Pro0, which is native from my homeland, Spain. And as well, we observed that there was a telomere shortening happening in LMS1 and LSP2, so different regolith simulants. So again, caution, genome integrity might be compromised in different ecotypes and in different regolith types. So I have to highlight that this work is being currently performed by 3 undergrads in the CIPEL lab, Emma, Cole, and Max, that I do not have his pictures here, so I apologize for that. So Borja, are we doomed? Is humanity doomed if we're trying to grow plants on the moon for multiple generations? So actually there's a technique that has been used for farmers for multiple times, which is reusing reusing soil, right? Reusing the soil to grow plants for different generations. And actually, that's exactly what we tested. We observed that reusing lunar regolith lead to improved plant physiology. So we observed that plants that were grown on reused LMS1 displayed a better germination, better leaf size, and an overall plant height compared with what we call a fresh LMS1, so a newly washed LMS1. And that's exactly what you can see here, that the earth soil plants are much taller plant height compared with the fresh LMS1. However, reused LMS1 rivals that height and is almost up to those plant size parameters of the earth soil. And here is just a very sad picture of untreated LMS1 Arabidopsis thaliana grown. So the question that we have, okay, what did change in this reused LMS1 molecularly that is bringing it closer to the earth soil? And again, we did observe that reusing lunar regolith led to a decreased genome oxidation compared with the fresh antioxidant regolith. So you can observe over here that the reused regolith grown plants have lower levels of genome oxidation compared with the antioxidant regolith. And potentially this decrease in genome oxidation led to the telomeres of the reused regolith grown plants to be similar to the earth control plants. So you can observe over here that they are still short, that they are statistically not significant compared with the earth control plants. However, they displayed a much better improved compared with the antioxidant regolith plants. So this work is currently being performed by another undergrad in the lab, Ethan. So I said a lot of things, a lot of data, so I just want to summarize everything. So plants that were grown on the International Space Station displayed as a telomere, no change in telomere length. However, there was a dramatic change in telomerase activity that we hypothesize it is due to increased oxidative stress when plants are grown on the International Space Station. So when we go to the moon to set up our moon colony, untreated lunar regolith will not lead to enhanced plant growth, and the plants will potentially die. However, treating plants with an antioxidant cocktail did lead to plant growth. However, plants display a telomere shortening, as well as an increased oxidative stress that is leading to a plant shortening. And things are worse on the next generation of G2 antioxidant. However, reused regolith plants displayed an increased telomere length and displayed as well a decreased oxidative stress and a much better plant physiological parameters. So there's a lot of people that are currently doing work in these projects. And first of all, we need to collaborate. We need to mention our collaborators here at Texas A&M, Julie Howe, Harrison Cocker, Terry Gentry, Jessica Atkins, as well as different collaborators that have worked for the flight from Ohio University, NC State, Universidad Nacional de Plata, Colorado State, NASA, Winston-Salem University, Florida Tech, Marshall University, and thanks again for funding for NASA and NIH. And if there's time, I'll take questions. And if not, just reach out and talk with me. Thank you.
Thank you so much, Borja. That was awesome. I think we have time for one question.
More excellent presentation. I want to ask you one quick question here. When you use lunar simulants, did you control the gravity?
Sorry, I don't
When
think.
you use lunar simulants for your experiment, because in the first part you did at ISS, right?
Yeah.
The second experiment, the one use lunar simulants, you just test the different material, or did you combine the gravity effect on it or not?
No, so for the growth with the lunar simulants, we did not do any microgravity associated with it. It's actually a very interesting question. We do have a collaborator in Winston-Salem University, Rafael Oreiro, that he is growing these Arabidopsis thaliana plants in lunar regolith simulants, applying the gravity that is specific for moon growth. So that 0.16 or 0.18 gravity as well. Another good point that you raised, all of these experiments, there's going to be some sort of radiation being associated with this plant growth, right? So ideally, one of the future experiments that we would like to undertake with this plant growth will be microgravity, radiation, and lunar regolith simulant growth. So I think that will be a more complete study to really, really dissect out what could be happening on the moon. But that's a great question.
Thank you.
All right, thank you so much. That was an awesome presentation.
Thank you.
For our last speaker, we have Charan GB, and who is a PhD student in Dr. Nithya Rajan's lab here at Texas A&M and the Department of Soil and Crop Sciences. Thank you for being here, and we look forward to your presentation.
Thank you, Harrison, for introducing me. I will quickly share my screen. Is my screen visible?
Yep, looks
Yes.
good.
Change the display setting. Okay. Howdy, I'm Chiranjeevi Porel, a PhD student in Department of Soil and Crop Science. The topic of my presentation today is real-time monitoring of plant stress hormones for extraterrestrial agriculture. And this project we are doing is in collaboration with the University of Texas at Tyler, and the research we are planning to conduct it in Texas A&M University. A beautiful flower picture at the background you can see in the slide is the first flower that was captured in International Space Station in 2016, and this is a zinnia flower. Moving towards the presentation, I think this introduction section is very familiar to all, but over here I would like to focus on the challenges that we are facing or we have to deal while we are doing the space farming, that are microgravity, extreme temperature, and limited supplies to sustain astronauts in long-term missions. Why we are doing space farming? Definitely for the fresh food source for astronauts for long-term missions. And it also provides psychological benefits, reduces the reliance of supply shipment from Earth. And this could be a very good opportunity for us to do research in microgravity condition. Current approaches, NASA have been conducting different research for plant growth in space station. The first one is the Advanced Plant Habitat. This project was started on 2011, I guess, but it was released on International Space Station on 2017. And this project contains the growth media as clay-based substrate. And the second one is vegetable production system, or VESI, as also mentioned by Dr. Deng. And in this project, NASA is specially producing edible vegetables, and this was launched to the space station in 2014. And it uses the growth media of calcined clay. However, in our study, we are proposing to use a growth media that is called coir peat or cocoa peat, that is made from the coconut husk. And this media will be compared with the regular medias that are hydroponic and peat perlite that we normally use as a potting media. Black-eyed peas will be our model crop, and we will be more focused on salicylic acid and abscisic acid monitoring in our study. We are developing a novel sensor that monitors those acid or hormones in a real-time manner, and we will also use like LI 6800 for monitoring different physiological changes that we can see within the plant. Why we are using coir peat? Coir peat is significantly lightweighted compared to the calcinetic clay or clay-based substrate that NASA is using. Currently, it is also very portable, that is transferred in a compressed brick form. Once this media is hydrated, it expands significantly. However, it still remains lightweighted due to the air trap within the fibers. And the main reason for using this media is because we can reduce launch weight that can reduce the energy consumption when we transport it from Earth to the International Space Station. And it also has excellent water retention properties. Why we are considering black-eyed pea as a model crop? Because black-eyed pea, or also known as cowpea, is an ultra short duration crop that matures in 45 to 60 days after planting. So we can have a multiple generation in 1 year or even 6 months within the space station. The second one is both the leaves and grains are edible from this crop. And currently developed cultivar for this crop are Busi, and it has moderately deep fibrous root system. And it is also day neutral cultivar and can be easily planted in the pot, and they are easy to plant and harvest in the pot as well as in the space station. Why and what control environment we are using? We are planning to mimic the environment or condition similar to International Space Station. For that reason, we are growing those in the controlled environment under different carbon dioxide and temperature levels. We will test the growth media and the crop in the ambient carbon dioxide level of 400 ppm and elevated of 700 ppm. We will also differentiate the temperatures with the normal temperature of 33 degrees Celsius for day and 22 for nighttime, and the elevated temperature of 38 for day and 27 for nighttime. We will set the photo period of 16 to 8 hours for light and dark. Why we are focusing on abscisic acid and salicylic acid? Because these acids or hormones are the key stress response hormones for the plants. And once these hormones are released inside the plant, it signals drought stress, extreme temperature, salinity stress, and oxidative stresses. Along with that, it also indicates some biotic stress. When the plant start facing biotic stress, it will also release these hormones. With the release of this hormone, there will be changes in the physiological parameters like stomatoclosure, reduced nutrient uptake, and plants will also try to cope up with the minimum oxidative damages when these hormones will be released. Sorry, I forgot to mention about this project. Initially, this project is in the development phase right now, and within these slides, I'm only presenting the methods. We haven't started, or we don't have any results to present right now. So from here, I will describe about the sensor, the novel sensor that we are developing in collaboration with the University of Texas, one of the electrical engineering lab at University of Texas at Tyler. This hormone sensor is the electrochemical based sensor that measures the hormone level in the plant sap. It quantitatively measures the salicylic acid and abscisic acid. The mechanism for measurement of those hormone is it measures the changes in the hormonal concentration by observing the proportional change in the impedance or the electrical resistance on the sensor surface. And this measured change in the impedance will be compared with the pre-built calibration curve that with which we are able to estimate the change or unknown concentration of hormones inside the plant sap. Talking about the accuracy of the sensor, as I mentioned, it is still in the development phase. However, the accuracy of the sensor seems pretty good with the R-square value of 0.99 currently when it is tested with the salicylic acid. As we can see in the picture, the sensor is deployed on the stem, and it is connected to the computer where we can get the concentration of the salicylic acid and abscisic acid easily from the software. This sensor is mainly optimized for the black-eyed pea or cowpea plant, and the concentration level it can measure till now is 0.1 to 100 micromolar. And with the use of multiple sensors at multiple points, we can easily get those data from the computer or the software. The main challenge that we are facing currently on development of this sensor is about its life cycle. We have to replace the since as I mentioned, it will be deployed to the stem. There will be a penetration needle that will reach to the plant sap, and at every week we need to replace those needles. So that is the current challenge that we are facing, is the reproducibility of the sensor for every week. Along with that, we will also use or measure different physiological parameter using LI6800 portable photosynthesis system from Lycor Bioscience. From this, we can measure photosynthesis levels, stomatal conductance, transpiration, and respiration. The mechanism of this instrument for measuring different physiological variation is by the differences in the carbon dioxide level concentration. Along with these parameters, it also delivers other different values that can be utilized for analysis on different plant status in other aspects. Lastly, regarding the coordination or integration, the novel sensor that we are developing for observing the changes in the plant sap, this will give us the real-time data of those hormones that can be used to inform a plant production and even alert the researchers or astronauts on the potential problems that is occurring during the plant production system. This real-time data will also facilitate us in automation of the plant production, and even with the when we incorporate this real-time data with the physiological data, then we can use these in the data science to develop a model from which we can predict the plant status with the real-time with the incorporation. Finally, with this, I would like to conclude my presentation and thank everyone for listening. And I would like to open the platform for questions, if any.
Thank you so much for your awesome presentation. I have a question about your sensor that you were utilizing on the cowpea. So is that sensor or is it inserted inside of the tissue, or is it clipped to the outside of the plant tissue?
It is inserted in the inside of the tissue in the stem so that it can measure the concentration in the plant sap.
Awesome. Okay, cool. So is that sort of like a thermocouple that you're reading that electrical impedance off of, or is this something that is actually like specific for these hormones that you're measuring?
Yeah, it is specific for the hormones designed for the salicylic acid and abscisic acid hormones. But it will have like a piercing needle inside it, which will poke in the stem of the plant, and by reading the changes in the electrical resistance or impedance level, we can observe the changes in the hormones inside the plant sap.
That's awesome. Thank you so much.
So how does the sensor specifically detect the salicylic acid and the abscisic acid? How does it know to? Because there's obviously lots of other things that are in there when you pierce the plant tissue. So how does it discriminate?
Yeah, that we are mostly the other lab, electrical engineering lab at University of Texas, they are working on it. But as far as I know on this, I got the information like they are specifically testing this in the flat salicylic acid and abscisic acid outside the plant surface, and also monitoring it with the different other acids, so that it will be more specified, and the calibration curve they are building based on which they can see the performance of salicylic acid and abscisic acid in the plant.
Okay, thanks.
All right. Well, I guess that's all. I apologize that we went a little over time today, everyone, but I think it was well worth all of the great research and presentations that we ended up seeing. This was a really exciting session, and we're super thankful for all of the speakers that were able to make it today, and all of the listeners who took time out of their day to come and learn. We went over some really exciting and very novel science that is being done today. I encourage anyone who is interested in this stuff to reach out to us. If there is, someone needs to find an email or anything, I think everyone can be found pretty easily, or I'm super happy to send out information. But again, I just wanted to thank everybody. It was super awesome. So